right, so today I'm introducing our speaker, Dr. Gracie Vargas. She is a professor and vice chair in the Department of Neurobiology and distinguished chair in neuroscience at UTMB Galveston, and she received her bachelor's in physics from Gustavus Adolphus College and a PhD in biomedical engineering from UT Austin. Her lab is the Advanced Biooptics Imaging Lab, and the title of her talk today is My Academic Journey in Light, Biomedical Optics in Non-Invasive Diagnostics, an Intravital Study of Neuroinfection.
Long title. Thank you, Kevin. Thank you for that introduction, and thank you to the Keck Seminar Committee for inviting me to speak to you today. So as you can see, my area of interest is in biomedical optics. I have an interest in biomedical optics and optical imaging for study of injury and disease, and then for developing non-invasive diagnostics. So my lab primarily is focused around, if I can move this forward, the use of nonlinear optical microscopy, namely fluorescence technique of multiphoton microscopy and a second related nonlinear signal called second harmonic generation microscopy for identifying image-based markers of disease. So many of you in the biological sciences may be familiar with multiphoton microscopy. It's as I said, a fluorescence microscopy technique that allows for deep tissue imaging. And it's based on the premise of using near-infrared photons that can excite fluorophores to excited state and induce visible fluorescence, where in essence, you are using two simultaneous photons with equivalent energy of a photon at its absorption peak to excite the fluorophore. And upon emission, you get, upon relaxation, you get emission of a visible light. Second harmonic generation microscopy is a related nonlinear process in which there is a frequency doubling of light. And the reason it's significant in biology is that certain non-centrosymmetric molecules such as collagen produce this frequency doubling with ultra-short pulse lasers, and it can be used for imaging. So this technique has been used in a number of areas from neuroscience to cancer and immunology for probably the last 2 decades. iLab is interested in using it in a label-free way and also in a label-full way for cellular and molecular imaging as well. So what we see here is a picture of a microscope that's in an upright configuration. Because this technique can allow you to image much deeper than traditional microscopy techniques, it gives you the ability for in vivo imaging. And the probability of fluorescence by this process is so low that you have to basically, in essence, prime the system such that you have an ultrafast laser and high numerical aperture objective so that fluorescence occurs only at a very small focal volume. And that's with the high numerical aperture objective. With laser scanning in an imaging system, then you can create optical sections and 3D imaging through raster scanning. So what you're seeing here in the image here are epithelial cells in the oral mucosa where the signal, can y'all see my arrow there? Looks like you cannot. Where the signal is autofluorescence from these cells. And I'll say a little bit more about that. So this term intravital optical microscopy is basically just microscopy in vivo. And it's been used, there's a number of different techniques that can be used for imaging cellular and molecular processes in the living animal. And then there's even systems that are being produced for various clinical applications. So the second part of what I'm going to talk about today, aside from the label-free imaging, is some of our work in neuroinflammation in looking at, in studying glial vascular responses in neuroinfection, primarily cerebral malaria. What you're seeing here is a CX3CR1 GFP microglia, or many microglia, with surrounding the vasculature in the mouse brain. So before I get into that, into more of the research, let me tell you a little bit about myself. So as Kevin told you, I'm a professor and vice chair for in the Department of Neurobiology at the University of Texas Medical Branch down in Galveston, just down the road. It was previously the Neuroscience and Cell Biology Department. And then before that, I was in the Department of Physiology and Biophysics that got merged into that. So if you're wondering how I ended up in neuroscience, that's how I ended up in neuroscience. I am the Cecil H. and Ida M. Green Chair in Neuroscience. I'm a fellow of the American Institute for Biological and Medical Imaging, AMB. You've already been told what my education is like, with a bachelor's in physics, master's in mechanical engineering from UT Austin, and PhD in biomedical engineering at UT Austin, with a focus in biomedical optics and biophotonics. So who am I? I'm also an educator and a mentor. So I am faculty in the Graduate School for Biomedical Sciences, and I mentor graduate students, medical students. I am a director of a post-bac research education program, and we train post-bac students from underserved populations to provide training for them to transition into PhD programs. And then I've also mentored a number of undergraduate and high school students through my lab. I'm an active and productive member of the scientific community. Produce scholarly work in intravital imaging, optical clearing, biomedical applications. And then in service, many of the things that you've probably heard from other speakers, reviewer on science for national science proposals, and conference chair. And the one I want to mention is the International Society for Optical Engineering, where I chair a meeting in quality and design. And one of the interesting things that we've been looking at in terms of having sessions is in health equity. So designing systems with health equity in mind. Okay. More on my background. So my family, I am a fourth generation Mexican American. And my family pretty much originates from the Texas area. These are my grandparents on my maternal side. This is Ismael and Maria Luna Salas. And they lived in the Victoria, Texas area. My mother, arrows pointing to my mom, is pictured here as an adult, as a young adult, with all of her sisters and one of her brothers. She had multiple brothers. So as you can see, this was a large family. This was a large family that was a migrant family that moved from place to place. And there was really no opportunity for education for most of them. Some of the younger siblings did go and get as far as the high school level. But for example, my mom had a second grade education. And pretty much she had to help others in providing for the family. My father's family was very similar. So my family, this is me right here in red with my younger sister. And when I grew up, I pretty much was raised by my mom. And by that time, my older brothers and sisters where I have 4 sisters, 3 brothers. As you can see, lots of nieces and nephews had grown and left the home. And so it was me, myself, and my mom. We moved around a lot. She was a single mom and basically just trying to make it. And so she would work for a couple years, struggle. We would then move somewhere else where perhaps we had family, more support system, and that sort of thing. So throughout my childhood, so although I was born in Texas, throughout my childhood, we moved places like Kansas, back to Texas, back to Kansas, back to Texas, Oklahoma. And then I think from Oklahoma, about that time I was in middle school, we went to Minnesota. And so I spent my high school years in Minnesota, so 9th through 12th grade. 2 different cities, and we did move with while we were there, but 2 years in one place, 2 years in the other. And I graduated from a town called St. Peter, Minnesota, which was an hour south of the Twin Cities. I was the first in my immediate family to graduate high school. Immediate and actually even extended family. I think my mom had some cousins that had, but in my immediate family to graduate high school, and for us, it was a big deal. So you see this big family here. Well, this big family is more recent, probably about 10 years ago, not when I graduated high school, but my brothers and sisters at the time were young adults, and they all made their way up to Minnesota. Nobody lived up in Minnesota, made their way up to Minnesota, and it was just a big deal. Now, what was I interested in at the time? For some reason, although nobody around me had gone to college, although no one around me had really said, you need to go to college, I always just knew I was going to. And I always just knew I was going in that direction. What was I going to do? I had no idea. I wasn't sure what my interests were. I had some interest in art, and I had a little bit of interest in science. So Minnesota is a state of what, 10,000 lakes and almost just as many liberal arts colleges as well. Maybe not quite that many, but there are many liberal arts colleges. And I was recruited by several of them and had offers to go to several of them on almost primarily, basically full scholarship. And what I ended up doing is I ended up going to Gustavus Adolphus College, which was in St. Peter, Minnesota, which I said earlier is where we were living. So that made it convenient because I could be close to family, close to mom to be supportive. And then but I lived on campus. So this was in Minnesota, and was it cold. But I had been up there already. I was used to it. College was an amazing experience. So I ended up in physics, and this is where I met my first mentors in a sense. The gentleman all the way to the left, that's D.C. Henry. He was the chair of the physics department. And after I took his physics class, he pulled me aside and he talked to me and said, hey, I think you should continue along this route. I hadn't made up my mind. I didn't quite know what I wanted to do, but I continued, and it seemed to work for me. This was a science building, Olin Hall, in which we had an observatory. And because we were a small school and we had some of these resources, it was really neat. I got to run this observatory, pretty much on my own with my colleagues in senior year. During that time, I did do a summer research fellowship at University of Pennsylvania. It was a National Science Foundation. This was my first lesson in what equity versus equality means, in a way. And we can talk about it in Q&A session if anybody's interested. But this was an outstanding group of faculty that were very supportive. And I think because it was a small environment, it fit me. However, I started realizing that I didn't know what I was going to do with physics as a career. I wanted to continue, and I didn't know that I would just go out and necessarily get a job. Towards the end of my studies, I started thinking, well, biology seems somewhat interesting, and could I bring what I know in terms of math and physics to the area of biology and impact health? I didn't quite know about biomedical engineering and those areas yet. I didn't quite have the mentorship in that sense. So I took cell biology and genetics class, which I probably don't remember as much about, but in my mind, I was putting physics and biology and saying biophysics, right? And I actually did start at a graduate school here in Texas, and it was in a biophysics graduate program. But I realized very quickly, but it wasn't quite what I had in mind of what I was thinking biophysics was. Which is funny though, because now some of the things that I do fit exactly with what the lab I had landed in does anyway. So I think I just wasn't at the maturity level or the knowledge level at that point to really know. But for me, it wasn't quite. So I started exploring, and short story, I ended up in biomedical engineering. I went to University of Texas at Austin. So I first started in mechanical engineering. Again, I was a bit of a lost soul. I knew I wanted something. I didn't quite know what. I was meandering a bit. And while I was taking classes in mechanical engineering, I took a course in light tissue interactions from Ashley J. Welch. And he was one of the early people that was studying light tissue interactions and kind of, the early originators of biomedical optics for tissue study and diagnostics and laser therapeutics. I ended up in his lab. The diagram here is, in essence, of depicting the photons, light going into tissue and being impacted by the tissue through scattering events, absorption events, and the signatures of the light that's collected back can be used to learn about the tissue. And so some of you may know that that these are used for different diagnostic processes, either through diffuse reflectance or spectroscopic assessment of the tissue. So anyway, AJ was a great mentor. My PhD was in a method. We were among one of the earliest labs to do optical clearing. We were interested in dermal applications because we were in a laser lab. So the title of my dissertation was Enhanced Penetration of Light in Biological Tissue, Implication for Light Diagnostics and Therapeutics. And what you're seeing here is a U.S. Air Force target on the left side. And in the middle, you see what is basically a thick piece of rat skin. And then after treating the rat skin, it becomes optically transparent and you can see through that. And so what I did in my thesis is we studied some of the light interactions. We looked at measuring the optical properties, and we found that what was happening was a decrease in scattering. And that makes sense. So it makes it transparent. And we think it was by an index matching approach. And then we explored areas for therapeutics and for fluorescence sensing and detection from deep lying targets. But my PhD experience, I already talked about AJ. He was a wonderful mentor and sponsor. He really influenced my direction. For all of us, he exposed us to many areas and had us collaborate with each other on different projects. We networked. He sent us to many conferences, so I had a lot of opportunities to know the leaders in the field. Friends and collaborators came from it, and it was a very supportive department overall. So I got a lot of awards, and these were kind of without trying. And I didn't know at that time that I was in a very protective nest, that the real world wasn't going to necessarily always continue that way. So the other thing I'll say about this is that my colleagues, my fellow students, you're sitting here with your fellow students, people that you're working with in labs or go to classes with. They're part of your professional network now, and you will potentially know many of them for the rest of your life. For me, if I look across here, there's a program officer at National Cancer Institute, an entrepreneur who started a company for an eye imaging company, another entrepreneur. He's a director of a biophysics division at the FDA. And basically these are my professional, this is part of my professional network, but also started off as my friends and colleagues in the lab. This was during a time, so this was early, this should be 2002. So this was 2 decades ago, I know. So in a time where there were a number of biomedical engineering programs and departments being started and centers. And so although I had only done a short postdoc, I was able to secure an assistant professor position at UTMB in the Department of Physiology and Biophysics. I was also a scientist in a Center for Biomedical Engineering, and I was hired to establish a research program in advanced imaging, primarily nonlinear optical microscopy, that I wanted to grow with applications in cancer, and that has gone to other infectious diseases and other areas. My optical clearing work, I got it funded by NSF, but I somewhat put it on the back burner while I got these other things going. So I established my lab first, junior faculty, and I established it as a lab to conduct research in advanced microscopy for in vivo imaging, using the techniques of multiphoton autofluorescence microscopy, SHG. We had a confocal reflectance microscopy set up, and then I was interested in instrumentation development. At my heart was this concept of light tissue interactions and label-free imaging. But of course, through collaborations, we also did more traditional imaging. Okay, so first I'm gonna go into one of the areas that I wanted to talk about. So I had an interest in developing an optical biopsy approach for epithelial cancer imaging, and using this multiphoton autofluorescence microscopy technique. So, and I'll tell you a little bit about that, but where we are with this now is that we're integrating, we're working on a device to integrate rapid large area surveillance by wide field fluorescence with histopathological scale multiphoton autofluorescence microscopy as the corresponding optical biopsy technique. And our goal is for it to aid in oral epithelial neoplasia detection to help in biopsy guidance in patients. So if you look here, this depicts the transformation. Again, I don't have the arrow, and I'm not good at this. The arrow. Is the arrow showing there? Oh, I should have done it there. I apologize. Okay, so this depicts the transformation that occurs when you go from normal to an invasive carcinoma, in which there's dysregulation and proliferation within the epithelium that starts at the basement membrane and moves its way upward until there is breakthrough at the basement membrane and you get invasion. And so there's a number of atypia that occur through here that include that pathologists look at in histopathology. They include abnormal cellular and nuclear shape and size, loss of basal cell polarity, loss of epithelial stratification, and then there's changes in the extracellular matrix and basement membrane remodeling that occurs as well. So dysplasia, this abnormality in these cells can be harbored in potentially malignant oral lesions that have a high risk of transformation. So not all cases of dysplasia will necessarily transform, but they have a high risk if they're moderate and high grade. So clinically, they try to find these and treat them as well as early cancers. But it's difficult to identify when you have heterogeneity within the oral mucosa and when benign lesions may also look somewhat like malignant lesions as well, or these pre-malignant lesions. And there's a motivation to be able to detect these changes because the majority of cancers are generally detected when cancers are in the regional and distal range, when the 5-year survival rates is below 50%. So the earlier that these can be detected, the better. So much of our early work was done in a hamster oral squamous cell carcinoma model, in which topical carcinogen listed here, DMBA, is applied topically to the buccal pouch over time. Controls have mineral oil, and then you can induce inflammation with SLS as well. And then in human tissues, we have clinically resected oral squamous cell carcinoma tumors. And then for imaging that I'll talk about today, we primarily use excitation at 780 nanometers. So we're primarily getting and collecting autofluorescence primarily from things like NADH, FAD as well here, along with, but we don't necessarily separate them in each case. And then these are some of the parameters that we've used. So this gives you an example of what I mean by label-free imaging. This is in vivo in that oral mucosa in one of those in the buccal pouch, in which you can see the stratum corneum with this cobblestone pattern of the keratin. Again, this caused by autofluorescence from the cytokeratins. In the epithelium, you can appreciate the nuclei in the cells and the very regular pattern that's here in this healthy tissue, and then the stromal collagen and given by second harmonic generation. In the case of dysplasia, there's dyskeratosis, so there's abnormalities in each of these layers. Nuclear atypia, where you appreciate the enlarged nuclei and the various size nuclei and cells, and then altered extracellular matrix in the stroma. And then this just shows 3D volumes from that. So after you do in vivo imaging, the buccal pouch is amenable then to histology. So you can take biopsy and then compare image metrics from your label-free imaging to histology. So the top is just color-coded. This would be the epithelium and the stroma. Here, same thing, epithelium and the stroma, and you can appreciate some of those, at least visual similarities. And then we use the combination of autofluorescence with the SHG in order to identify the epithelial connective tissue interface. So in essence, that basement membrane layer. And when you're looking at it in histology, you're seeing it in cross-section, but really you can think of this as a 3D sort of sheet. So this is a case of healthy mucosa, and this is one in which there's that dysregulation that's happening in oral epithelial dysplasia. We've also used microspectroscopy to capture where the source of signals is coming from And we're primarily looking in two ranges, in the red region with the porphyrins, and in the green region that I mentioned before, where you may get NADH fluorescence as well. And then using a combination of fluorescence from NADH and FAD, one can calculate the redox ratio. And this is a method that a number of people in the field are using. So we have used the method for doing quantitative in vivo cytology and looking at atypia both in the epithelium as well as in extracellular matrix. Here's an example of a parameter we call the coefficient of variance of nuclear area, and is somewhat akin to anisonucleosis, that pleomorphism that I talked about. And what's significant about this is that in our model, we always include inflammation, and inflammation clinically is always a potential confounder. And because you don't necessarily have the same microstructural atypia, we can differentiate dysplasia from both control and inflammation, or healthy and inflammation. And then similarly with the collagen density, which decreases. And then using a graphical representation of a binary classifier, of which are these classifiers, at varying threshold levels, the receiver operator curve characteristics, we can look at the total area under the curve and get a feel for sensitivity and specificity based on using these. And what the main thing I want to point out here is that the green curve is that of just comparing a control versus dysplasia, whereas the blue curve is grouping these together against dysplasia, and we see that the performance is still very high. As I told you, we can get human oral squamous cell carcinoma tumor samples as well, and these are examples from various anatomical sites of the tongue, the larynx, the floor of the mouth, in benign conditions, dysplasia, and squamous cell carcinoma. And this is that parameter, coefficient of variance of nuclear area, and it has a very similar trend as we found in the animal model that I had just shown you before, in this preclinical animal model. And then one can do visual classification of features as well. I don't really have time to go into all of those. But through a lot of collective work, we and others have been able to show that multiphoton microscopy and epithelial neoplasia can provide image-based markers for a variety of cancer hallmarks that include dysregulated cell proliferation and differentiation. Those were some of those cellular features that I talked about. Altered metabolism, such as the redox metabolism, and then altered protoporphyrin IX peak for heme metabolism alterations, basement membrane remodeling and pre-invasion, and extracellular matrix remodeling and pre-invasion. And these, I differentiate this because this is the ECTI versus the deep stromal tissues. So there's some promise in being able to use this, and that was our goal, was to establish feasibility for being able to detect cellular and extracellular atypia. But the problem with this technique I told you at the beginning is the microscopy technique. So how do you really scan the oral cavity? So what we've been pursuing is a combination of techniques that have already made it to the clinic, and many decades worth of work from investigators, including some here at Rice, that have established that you can use large area fluorescence by blue light to induce autofluorescence and then look for loss of autofluorescence that may highlight areas of abnormality. So the nice thing about these is that this is a Bell scope down here at the bottom, the clinical device. The nice things about this is that they give you large areas of up to centimeters for rapid surface assessment, and they're highly sensitive to change. However, they're very limited. They can be limited in specificity. I told you before, inflammation, for example, is a confounder, and other benign conditions are a confounder. But they have been shown to be useful for identifying precancers in some studies. And then on the other hand, we have the technique I've been telling you about, multiphoton. I've color-coded it differently here, where the epithelium is in purple and the underlying stroma is in green, where you can get these where one can extract cellular and extracellular atypia. So we've studied workflows for how we can combine wide field fluorescence with multiphoton here in a benchtop system for wide field fluorescence. And we and others have been interested in collecting not just the green autofluorescence, but also the red autofluorescence from the tissue and extracting that as a wide field parameter and being able to highlight areas that we deem as potentially suspicious, conduct multiphoton imaging, and then conduct analysis for feature extraction, and then with histology always serving as a gold standard. And so within these, we have been able to look at the combination of the wide field fluorescence parameters together with some of the parameters from multiphoton microscopy with large area under the curves as well. And then some very preliminary multivariate models where the clustering shows separation between groups. And so what we've been working on, we then moved on to this concept of integrating both of these techniques into a single probe, and that's what we're working on now. And I'm working with two labs, the Rangquang Liang Lab at University of Arizona. Rangquang has expertise in mobile-based intraoral camera imaging, including with fluorescent. His background was in bright field, and then he's moved into fluorescence. And then the Jingdi Li Lab has expertise at Johns Hopkins, has expertise in miniaturizing optical technologies that include optical coherence tomography and then two-photon microscopy as well. And so the three of us have a multi-PI project that we're working on now, and we're collecting. So we realize we don't have the best images of the device here, but here is the device that we've built, and we've been conducting studies on it on one of our traditional microscopes as well. And you can see its components here. But anyway, this is in process, and so I don't have much more to present on that. And I'm going to switch gears real quick and get back to the other thing I told you I was going to talk about today. So I told you I started off in optical clearing, and then somewhere along the line, we did go back to doing some optical clearing. And what this shows is, apologize, it looks like my battery's running out. This might not have it. Okay, and what this shows is one of the techniques that we use for clearing mirroring lung. So this is the lung, and this is it after it's been, in essence, treated with a chemical agent for clarifying it. And we've applied this for studying. Okay, for looking at airway inflammation. So here is a multiphoton autofluorescence microscopy image of the mirroring lung, at least two lobes of it, where the green is the second harmonic generation signal from the collagen, and the red represents that autofluorescence. And then we've applied it for fibrosis. Well, these efforts in optical clearing actually led us to a collaboration in neuroinflammation. I know it's not the lung, where we began applying these methods with a collaborator, Robin Stevens, who was at UTMB and is now at Rutgers, who wanted to look at the brain and wanted to look at coagulation in the brain. And so this was a project in malaria, and specifically cerebral malaria, which is a febrile encephalopathy that affects over 500,000 people annually, and primarily affects children in sub-Saharan Africa. It has a 15 to 25% mortality. And as I said, we were interested in coagulation, and particularly fibrinogen-driven neuropathology in malaria infection. And this was because through optical clearing, we did, and other multiphoton imaging efforts, we did an early study to look at the presence of fibrinogen deposition in a cerebral malaria model, where we demonstrated in a hyperinflammatory experimental cerebral model that there was significant fibrinogen deposition within the brain of animals versus uninfected wild types. When we use low molecular weight heparin, that has eliminated the thrombi, and it rescued the animals without a change in parasitemia. So this then prompted us to ask, what are some of the dynamics that are occurring within this, and could we study this system in vivo? So what Olivia Solomon, who just actually graduated this last summer from our lab, worked on is on the dynamics of vascular dysregulation, primarily looking at the coagulation time course and its effects in the brain. And then we had an interest in the glial vascular component of that. So if microglia are the immune cells of the brain. They're the immune surveillance of the brain that have a number of different functions that include synaptic pruning, phagocytosis, myelin turnover, and have some role in neuronal excitability as well. Their morphology, just pointing it out because we're going to talk about it, goes from a small soma ramified morphology to one in which you have hypertrophic processes and an amoeboid appearance when they become reactive to their environment. And I said it earlier, but the way that we're imaging in the brain is through a cranial window. Actually, it's a thin skull preparation where you use a dental drill to thin down the skull, and then you can image the upper layers of the brain, the upper, say, 250 microns of the brain. We have been studying two different models. One is a very common model of cerebral malaria called P-Burke-Anka in a CX3CR1 GFP model. And the other is a P-Shabodi model in an IL-10 knockout CX3CR1 GFP, CCR2 RFP model. And so after infection, we can follow the animal with behavior, and then at time points for imaging, either do imaging, behavior, and parasitemia for correlations between those. And we use the behavioral score to assess when they've developed cerebral malaria. So what I'm showing you here is a movie in vivo, and I'm not going to be able to show too much. I'm looking at the time, but of a case in where we are using IV injection of fluorescent fibrinogen in order to see where the thrombosis occurs within the brain, and then looking at some of the dynamics. So if you look at this movie, I don't know if you were able to catch it, but the green represents the fibrinogen that is within the vasculature. And if you can tell, you can see some flow, and I don't know if you can tell from where you are, right here. I think my little bar is kind of covering it, but you see that there's a little bit of flow, but the flow has stopped here. And then there's instances of some perivascular puncta that are out here in green that are perivascular fibrinogen. And this is another sample where we can see the perivascular component of the fibrinogen. This one is a C57B, so there's no GFP in there. We're confident that that is the fluorescent fibrinogen. When we compared across models, we found that both P-Burke-Anka and P-Shabodi displayed very similar thrombosis characteristics. You can see the green fibrinogen in both of these cases, and in both cases, the perivascular fibrinogen. So I told you we're interested in some of the dynamics, and this one shows an example of the dynamics that can happen. So we're giving it IV injected fluorescent fibrinogen, but of course you can have formation of thrombi that maybe are endogenous. And so what we have here is an occlusion that's keeping the blood from flowing, and then we have a microhemorrhage that occurs. And then the green is the fluorescent fibrinogen. So the advantage of intravital microscopy is exactly this, that you can look at the dynamics and potentially study the dynamics over time and over time of infection. These models, most studies look at these models at the very terminal time points. And that's where we started and are moving our way to earlier time points. Whereas in histology, you can see that there was a microhemorrhage, but you may not necessarily know the events around that. And where that becomes significant is in things like the microglia that I mentioned before. So what you see here in this dynamic image down here is a microglia that's close to a vessel in which there's a thrombus that's about to form. And you can see the change in morphology. These are 2 different time points. The change in morphology that occurs as this forms. And you see the process is almost moving towards the vasculature. The microglia, in this case, we have CX3CR1 microglia. And what I think you can appreciate is that the soma, these are the same scale, the soma become enlarged. And then you see this co-localization of the green with the red that is occurring here in the microglia that are up against the vasculature here. And so we see that in infected animals, we have, I'm calling this a glia score, which is just a visual assessment of the reactivity for now. And then the vascular score, which looks at changes in flow, leakage, and those kind of aspects. So if we look at the makeup of the microglia, we find that there's much more heterogeneity that occurs in the experimental cerebral malaria, where initially you start off with primarily ramified surveying, as you would expect in a healthy animal. And then after this, in this case, this is day 7 of the model, and you have the majority of them are hypertrophic or amoeboid. So you appreciate that they're very reactive. And as a surrogate, we've used, oh, did that not come up? Okay, soma diameter for image-based measure for that, in addition to things like shape parameters, a transformation index for looking at reactivity. So what we've pretty much, the short story, because I can't present everything, is that in addition to these reactive microglia, we find that they then move over to the vasculature. So the vessel-associated microglia increase substantially over time. This is at the late time point, and this is the mid time point at day 3. There's not a significant difference. It looks like it's trending, but it's not significant. And then, so those microglia move over to the vasculature. And then the second component is looking at the interaction with fibrinogen. So fibrinogen microglia can bind fibrinogen to CD through a CD11B dependent binding. And what you can see here is a phagocytizing of fibrinogen. You see the yellow phagosomes within the microglia as it's sitting here surveying. And the fibrinogen is either contained within the vessel or has come out into the perivascular space. And then this can be quantified as well, where there's an increase in fibrinogen engulfment by the microglia. We did look at what was potentially pulling the microglia over to the vasculature. But the thing that I wanted to say is that when we then deplete the microglia using a PLX 3397 chow, we get substantially increased coagulation. And so we think that the microglia, which might be expected, are playing a protective role in being able to process the fibrinogen. But we don't know how, in essence, the details of that from within the intravascular space, the extravascular space, and the time course of that. So this intravital imaging is giving us the opportunity to study a lot of the components, and there's opportunities to continue to study the components of dynamics of how the microglia are interacting with the vasculature, look more at the vascular dynamics, and there's a whole nother component around the vessel component that I haven't talked about, but that we can continue. So there's a whole lot of opportunity for studying the system and looking at, I said earlier, for example, microglia have a role in neuronal excitability. That's another area that we've started collaborating. Robin has started collaborating on these areas as well. So you kind of get a feel for the fact that my lab is broken into different arms in which we primarily are centered around the technologies. And while we don't jump all over the place necessarily, there are general areas of in which collaboration may lead to our interest. So I told you about the disease detection and early detection of epithelial cancers already. We talked a little bit about the study of disease through infectious disease and neuroinflammation. We have a glioblastoma project. And then our cerebral malaria is taking us in the direction of the retinal brain axis as well. And then I didn't talk about this, but we have an entire area in fluorescence endomicroscopy for studying epithelial injury, also in vivo, and in this case, it's in women's health studies in cervical vaginal for HIV preventative agents. So the results of my PhD, I think, gave me the flexibility and the ability to be able to integrate well across different scientific areas, to be able to interact with scientists in other fields, and applying our approaches to these areas. And hopefully through this advancing the use of optical imaging in biology and medicine as it is growing. We've been a highly multidisciplinary research program, and as I said, across a lot of different areas. My early work, it's very nice that we were able to work in this area of optical clearing, which actually was a field that has grown tremendously over the last 20 years. It has been very, very fun to watch. It's been incredibly rewarding experience from many aspects, and one of the most rewarding has been that in mentoring and being able to work with graduate students, undergraduates, the post-bacs that I told you about through our training programs, and be able to just simply to be able to mentor. It's been one of those things. And throughout my career, being able to benefit from the mentoring that I got. AJ Welch I mentioned, but throughout my academic career, as I went from assistant professor to full professor, there were certain people that served as either mentors or sponsors. The prior dean of the Graduate School of Biomedical Sciences, when I went to him and I said, hey, I've heard that there's this prep program and I would like to get involved. And he made a space for me immediately, and it didn't take very long for me to become very integral within that program. So sponsors like that helped throughout my career have been really important. And I think I was asked to give kind of some thoughts about my experience in STEM, and I didn't quite know what to hit upon. I guess I have some items of advice that you can take or not take. But for me, I think that what's important is that you do what you love, something that you really enjoy what you're doing. I told you about my career path in which I went certain areas. I realized that this was not the area for me at that point in my life. And I backtracked and went to different area, and I haven't regretted it. I've been happy with that. So and within my research, I followed a path based on my interests as well, trying to keep focus within certain things, but also trying to follow the things that are of interest and help our research program. Make sure you believe in yourself and follow your instincts. That's something I learned early on. Some of the ideas that I had as assistant professor, some that I didn't follow through, some that I did. I've learned that really trying to get, follow your instincts along, of course, with taking in some mentorship. Don't be afraid to take some of those chances. Be flexible and adaptable. And then when your confidence wanes, because I think it happens for many of us, just be gentle with yourself. Don't expect too much of yourself. And don't forget to celebrate the small victories, because one of the things that sometimes I forget is those small victories are the things that get you to that big victory. So when you look across and you try to compare yourself to somebody else, you're like, wait, I don't have that, that accolade or whatever, but down the road, then you can potentially have that through those small steps. And it's how it's happened for me anyway. Find good mentors, but also be a good mentor. You can be a mentor to your colleagues as well. That's another thing that I see all the time. And then on the flip side, recognize your biases and try not to let them get in your way, and recognize what your I'm working on list is, and be conscious of it and continue to work on those things. For me, I'm terrible at networking and negotiating. I've had to work on it quite a bit over time. I'd never felt like I had the mentoring, or there were always secrets I didn't know. I also come from a background and culture in a way where if you tout your successes, then you're bragging, and it's a negative. And so it's been one of those things that's been very hard to fight, to not, get involved with to get involved with kind of the PR side of science, but that's required. We need to be able to tell people what we do. And then always confidence building. Okay, and put in a plug for our program. Prepping for Success is our post-bac research education program. It's a 1-year research intensive program in which post-bacs are placed into a lab and conduct research for a full year. And they go through career and skills development and apply to graduate schools. And hopefully the next fall, they're getting into a PhD program in the sciences. We've trained 65 scholars, and while I've been involved with 43 scholars over the years. Okay, and then there's many people to thank and many different across the various projects, and hopefully I didn't forget anybody, but thank the people that were central in all of these projects that are listed here. Any questions?
Any questions?
Hey, really great talk. I'm just curious with some of these imaging methods in like, if you're moving them towards a clinical setting, what kind of the actual depth of field that you can actually get into tissue? Like I understand with the animal models that like you might be able to see the entire brain, but if you're taking this to a human scale, like
Right.
how deep into the sample can you actually visualize?
Right. So for clinical, we're primarily talking about the epithelial space, not necessarily the brain, because you're still talking about hundreds of microns. Now, there's recent developments that are pushing the limits to still not deep compared to whole body imaging, but millimeters. And now the big thing with multiphoton microscopy is 3-photon microscopy. So using instead of 800 near infrared 800 nanometer light or to 1,000, perhaps using 1,700 nanometer light. And where we're talking about that max is probably like a millimeter to 2 millimeters. And so it's still epithelial in nature. So what was really promising for us was that in the epithelial imaging project, when we took samples from the resected tumors, we always image from what would be considered the surface, the mucosal surface. And that's the geometry that we use. And we're still getting, even from those superficial depths, being able to detect some of detect the cellular and atypia that I was talking about.
Very cool. And then a kind of quick follow-up as well. You're able to distinguish between kind of individual cells, but like what is the kind of global resolution of these imaging methods?
So, okay, so when we're talking about the benchtop, you are at the micron level pretty much. With our device, we are aiming for about a little over a micron. Right now, we're optimizing it. We're at 2.5 microns lateral. Mm-hmm.
Awesome. Thank you.
Mm-hmm.
Anyone else?
Again, it's been too long since you've given a seminar. So the latest seminar here recently, evidently this fad is 3-photon microscopy.
Right.
So when I went to Coherent to ask about upgrading my 2-photon laser, we got in
Right.
2003. They want to sell you the 3-photon microscope as the latest fad.
Right.
Have you thought about that or evaluated it? Is it worth the money or is it just later?
So absolutely. So, so it requires an OPO laser, right? And so the cost is the main thing. And however, a number of the microscope companies have already optimized their paths such that you can implement the 3-photon. So it's not going to be as hard as, say, in the old days when people were trying to take confocal microscopes and create them into 2-photon microscopes, because now you were in a completely different wavelength range. You are in a different wavelength range, but for a number of years, microscope companies have already been working to push the limits out to further and further into the near infrared. So at least from what I'm seeing at the tech shows that there's a number of companies where logistically, it's fairly straightforward to add the 3 photon, given it's a completely different laser and wavelength. But yeah, it's the next thing that we would like to implement in our lab as well. But we have not yet.
I think I heard surgical pathology is moving in a digitalization movement. Do you have any collaborations that or have you ever dwelled upon reaching out to surgical pathologists and pitching these methods to them?
So, yeah, so we've worked with pathologists for a number of years as a pathologist as part of our team, but not anybody that's necessarily looking into those areas. There are some groups that are moving into that and working more on developing kind of these as pathological tools, as tools for pathology. And then, like you said, in terms of the methodology, I think that there is room to go in that direction.
Okay, these are beautiful pictures.
Yeah.
Beautiful work also. I was just curious, is there a way to turn this into, I guess, raw data?
Right. So machine learning and AI is, an area of exploration. In fact, Paula Villarreal, who's here, I think I showed her picture up there, is one of the things that she's exploring within her thesis as well. So you're right. There's much so that you're not always restricted to image processing algorithms. There are alternatives to that, and that's what Paula will be exploring.
Okay, well, thank you very much.
All right. Thank you for having me.